All right. And we are live with May here and Magnus. I'll get back to you guys and thank you guys for joining online. We're here with a teaser of the experiment designer, which we're very excited about. I just have some I came prepared with some slides, and I'm just gonna pull them up here. Oh, intro slides. The product teaser, the experiment designer. May Magnus, who are you guys and why are you here?
Shall I start?
Yes, good.
My name is Ame. I'm a product lead here at Reshape. I essentially see my role as twofold. On one hand, I am making sure that our entire customer experience across all software and hardware products is as seamless and easy as possible. But then I'm also very much always looking for new ways to solve additional problems and make the whole experiment analysis and execution workflow as easy and seamless as possible.
Good intro. I'm Magnus, and yeah, a bit more on the customer-facing side. I work with a lot of our customers and the team at Reshape, rolling out things, implementing, validating, and I also lead our internal application team. So I get the pleasure of typically being sort of the internal customer and working with a lot of exciting stuff we launch. So yeah, so excited to see what's coming out today.
All right. And I hope that everyone out there has sound. At least I get internal notifications that we have. Okay, Adrian, thank you. Good that you have sound now. And now that we heard from you guys, I just realized that I didn't introduce myself. I'll make it short. My name is Mads, and I do marketing here at Reshape. And today we're gonna be talking about this guy, the experiment designer. You guys, you wear some, Magnus, you wear a lot of different hats. Choose whichever one you want and talk about why and how you're excited about the experiment designer.
Yeah, for sure. I think I'm going to take the internal customer hat, and I'll start by shamefully admitting that even at Reshape, we still use a lot of Google Sheets and even paper notebooks and struggle with experiment design for a lot of our internal stuff. I think it's a difficult problem to solve, so we talk a lot about it internally. I'm really excited to hopefully be able to migrate that into the product. I think, yeah, on the data acquisition side, it's been super nice. We have a lot of devices here, but I think sort of like scaling up how you design experiments has really been a continuous struggle, right? So I think that's my number one thing, right? I think it's a big unblocker.
Yeah, I can only echo that. But being a big part of also conceptualizing the experiment designer, I'm also just very curious how it's going to help people in their day-to-day lives. I'm curious to see if there's these more complex experiment setup or people trying to miniaturize their essays, if this could be a very helpful tool in that, because we know it can be a daunting task to plan out experiments, especially if you do it in Excel, managing all the metadata and all the results in Excel. It can be daunting. So we're hoping to make that threshold a little lower and see if people can get to these things more seamlessly.
I think we have a few of those very scary Excel sheets. So hopefully that's going to be one of the things.
We'll try to eliminate those. All right. So just a quick agenda slide. The welcome that we're doing now. We're gonna run you through the lab of the future, what we're essentially trying to do at Reshape. Then MA will warp her to the product presentation room and talk a little bit about our latest and greatest feature launches. And then, of course, we'll come to the experiment designer. We'll round everything off with a Q&A. So in the chat, Adrian, Toge, you already know how it works, but write up your questions and send them through, and I'll try to moderate the Q&A in the end. Our colleague Jim is also on the line, so anything that can be answered fairly fast, he'll do his best to answer it as we go along. But without further ado, we're going to warp you up to the product presentation room and yeah, talk about the launches that we recently did and, of course, the experiment designer. So, May, take it away.
At Reshape, we're on a mission to build the lab of the future. Reshape is transforming how experiments are designed and run, bringing structure, traceability, and automation to lab work. We've come a long way doing this for analyzing your experiments through taking images automatically detecting all the objects on the plate and giving insights. But we also are working on a whole new side of our platform that helps you with design of experiments. But before you see what we've been brewing, I give you a few product highlights from things we've recently released. One of our big themes at Reshape is building to scale. We will always be finding the next thing to build, inventing new things, trying out what works in labs and what doesn't. But we're also committed to keeping Reshape equally easy to use with 100 devices and 1,000 users in your organization. A core element to facilitate this is our API. The API helps you keep systems in sync, your LIMS or ELN, and Reshape. You can send core experiment entities like media, treatments, or organisms to Reshape. And in the other way around, you can take your experiment results from Reshape and store them directly in your LIMS or ELM if that is what you use as a source of truth through our API. And this means that if you make any changes in either side of the system, those changes will reflect and everything will be in sync at all times. Another thing we've worked on is to help make it a little easier to run experiments if you have Reshape running in multiple labs in your organization. While usually most of the work happens inside a lab and with the people assigned to that lab, we also acknowledge real world is messy. So you might have other teams sometimes needing access to your devices, or people coming by on temporary assignments or even traveling and just wanting to shortly have access to your devices in a lab. This update allows group admins to quickly assign the right access to the right people at all times. Another important theme for us is unlocking deeper insights. We will help you make sense of your data by identifying patterns, outliers, and trends. And a core thing we've done here is to add the ability to add more context to your plates. So of course, we can look at a plate and identifying very kind of technically what we see, but the context is what makes it interesting. So you can now store things like your sample ID, your control type, what organisms you expect to appear, what media you used, what treatment you're testing. All of this information you can add right in Reshape. So also when you look back at historical experiments, you can quickly see what parameters you've used and what you're actually looking at when you see images of a plate. Another thing to help ease collaboration is our activity log that we're now showing on jobs and wells. So whenever something is changing, maybe you're collaborating on an experiment, whatever is changing or whatever happens, you will always be able to go back in the timeline and see what has been going on. Lastly, an important thing for us is to build trust and probably continue building trust in AI analysis. AI is starting to be all around us, and probably I'm not the only one that sees it goes wrong every once in a while. And it's important for us that you can trust the analysis we do and have it validated against your own expectations. That's why we built analysis validation. It's essentially a two-step process. First of all, you need to input your manual counts. So there's an easy workflow in our software interface where you can select the plates you want to use as your internal validation set and then quickly add the manual counts. And this can also help us understand your interpretation of a plate. Maybe you only want to look at the bigger colonies and not the very small particles. So that helps us set a ground truth for your expectation that we can then benchmark the models against. Once you've gone through this flow, you'll be able to see the overview in a quick performance overview. This is also where assessment from multiple people can come together and where you can explore outliers live. So you can click into the plate where there's a discrepancy between the human count and the analysis count, the automated analysis count, and see, is it the analysis that missed something, or is it actually the human that just looked at a snapshot in time, counted a certain amount of colonies, but maybe if you look at a time lapse, you actually see there has been more things growing over time. All of these things are super easy to explore live, but also get an overview of the overall performance of the models. That was it for the recent launches. Now I'll show you a bit what we've been working on in the new side of our platform, the Experiment Designer. For a bit of context, if you look at the experiment process all the way from design to insight, we've been quite heavily focused on the latter half of the process, from imaging plates to analyzing plates and getting the insights. But we also believe that the real key to automation is bringing this entire process together in a seamless flow and making sure there's no gaps and no data is lost between any of these steps. That's why we aim to support the full process. And today we'll talk about the design of your experiments. We've noticed a few common pitfalls when it comes to experimental design. We also have a big Giga Lab right above our office, where we can try a lot of things. And what we've seen is that human error compounds quickly. When we plan out an experiment, for instance, in an Excel sheet, it only takes one wrong dragging of a cell to suddenly result in being surprised about things growing in your plate, losing track of what you actually put in, and the results of that, all that hard work being meaningless. Another thing is that offline simply isn't enough. It might be okay to keep track of what you're doing in the lab in an offline way, but if you then later on get the analysis of your plates and you have that context of what is inside the plate stored somewhere offline, it's very hard to actually get the insights you need. And finally, there is an issue of prioritization. As scientists, I think everyone's curious, everyone wants to learn more. It's easy to think of more parameters to bring into your experiment, but we also all bound by limited resources and time. That's why important, and we want to help you make sure that you prioritize the parts of your experiment that have the highest chance of actually bringing you valuable results. I'll now show you what we've been working on and dive into a version of our product that we are working hard to get live and bring to you as soon as possible. All right, let's have a look at how we envision the experiment designer to work. And for those of you that are already familiar with the Reshape platform, you can think of an experiment as essentially a collection of plates or jobs that you need to run to reach the conclusion of what you want to learn. In this case, I'm going to run a fungal inhibition assay. And as a description I put in, I want to assess compound effects on fungal growth to identify optimal antifungal candidates. Now I first need to select the very basics of my experiment. I'm going to select an analysis type, fungal growth inhibition. And this is essentially the model that's going to analyze the images that are coming in. And I've already validated this in a tool I've showed you this before. So I know it's going to identify the right objects, measure them correctly, and give me the right metrics as output. Now I'm going to select a target metric. In this case, I'm most interested in the growth rate, and I want it to decrease. I'm still getting other metrics as well, like count or area measurements, but this is just to help guide the visualization downstream to reach my conclusions as soon as possible. I'm also going to select a key parameter, and this is essentially the thing I want to vary across the experiment. And in this case, that's going to be treatment. And treatments can be lots of things. You can see here, you can add the compounds, formulations, or samples you want to test. So in this case, I'm going to add some antifungal compounds. So first of all, I'm going to select vanillin and add a second and a third compound as well. You can then define the application settings either globally for all treatments or make them specific for each treatment. In this case, I'm going to select 3 dilutions and have a volume to put in each well. I'm also going to add the organism because I know what I'm going to put in. You can also leave this empty if you are not sure of the details of your organism. And then I'm going to define 2 controls. I want to have a positive control and also a negative control in each plate. Regarding the setup, I'm interested to have 3 replicates. I'm going to select which media I'm going to use. And here you can see I'm getting an AI suggestion to use potato dextrose agar because it's ideal for fungal growth and antifungal testing. And then finally, I want to run this experiment in a 24-well plate. All right, let's have a look at our run conditions. So how I actually want to incubate and image the plates. This is a standard setting for each job that you can make in this experiment. You can also vary it down the line. We'll look at that in a bit. It all looks good. The only thing I want to set here is my target temperatures that I want to test at. You can see that 30 and 37 degrees are suggested. I'm going to just go with those suggestions, but also add a third temperature at 25 degrees. Okay, if I now continue, I'm going to have an overview of those 18 plates that are needed to reach my conclusions based on the parameters I've just defined. And here I can just have a look at the layout of each of these plates. So I can see there's the 3 replicates, there's the dilution series, and there's the controls in the bottom. And then each plate has, of course, a different content that I'm interested in. So this all looks pretty good. If I'm not happy with the layout, I could edit the plate, but in this case, I'm just going to approve it all and continue. So now I'm done setting up. I just need to review that everything looks correct. I also get a summary of the preparation needed for this experiment, how much I need of everything, so I know what to prepare in the lab, and then I can create the experiment. So here we are seeing the overview of the experiment and what plates need to be run. You can also follow along on the timeline and progress here on the right. And this is all the plates that need to be prepared, incubated, and imaged. I could also click into these plates and go back to the view we just saw before. So when I'm in the lab preparing the actual plates, it can be a useful guide to see what goes into each well. For now, I'm just going to select all of the plates that need to be run at 25 degrees and create a new job. Essentially, the thing I need to do here is just select the device that's free at the moment, confirm that all the plates are in the right slots, and confirm that the run conditions set out in the experiment are also good for this job. That all looks good, so I'm going to start the capture. Now this would normally, of course, take a while, but we're just skipping ahead a day or two where some growth are starting to appear in the wells. This is an overview of all the 6 plates I've decided to run, and I can click into each of these. Here we see an overview of this particular plate with all of the wells. And because we gave all of the parameters and assigned them to each well, we're already getting to see some first conclusions regarding this plate and how the different treatments are performing. Now, if I see an interesting well, I can also click in there and analyze the metrics of this particular well. And what's useful at this point is you can also go into the details of the well and see exactly what the contents and the parameters of this well are. This can be useful both for collaboration. If a colleague comes in who hasn't been part of the planning and the preparation of this experiment, they can still make sense of what they see. But it can also be a great help if you come back to this job maybe a year from now. You're looking at the growth and you want to see what exactly were the parameters I used back then. But of course, it's not about the individual well. We want to reach conclusions about the overall thing we're trying to assess, how the different compounds have effect on fungal growth. So this is where the overview of the experiment comes in. We can see we're nearly done, so most of the conclusions should be there. It's probably just a matter of building up some more statistical significance. But we can see at this point, vanillin is the top performer. And here you can see how the growth rates are at different concentrations for the compounds we put in. So this should make the entire process of having a hypothesis, knowing which parameters you want to test, and actually reaching that conclusion as seamless and as fast as possible.
All right. And we're back. We've warped MA back down. Thank you. That, I of course think that looks very exciting. But this is sort of a greater vision of where we want to take the experiment designer. Could you talk about what are the timelines in getting this live in front of our customers?
$1 million question.
Yes.
Yes.
When can I buy?
We're working hard on developing it at the moment. We're expecting a beta version to go live in the summer. And what we want to do with the beta version is, on one hand, we want to bring this into the real world as early as possible, because it all works nice and well on paper and in theory. But I think we're really starting to see the effects once we use it for real. So we want to do that as early as possible. We, of course, also want to provide a great experience. That's why we are probably going to aim at supporting a somewhat well-defined assay type to begin with and just test that out. So that's also why I'm super curious to get in touch with people who are keen to try this and learn about their specific needs and kind of focus in on the first version to support. But yeah, first version to go live in the summer, and then we'll iterate from there and more and more will go live over time.
We saw the fungal growth assay, but do you have anything else lined up in terms of what types of assays it will work for?
Yeah, so the fungal growth inhibition assay, that's just an example. We made it the very structure of an experiment to be comprised of treatments, which you could see in the video as well, is quite a broad concept, and then organisms and media. And we expect a good amount of assays to fit that very core kind of architecture. So in that sense, we'll be able to facilitate running a lot of different types of assays. But of course, we also need to allow the right type of configuration. Maybe there are some more customizable things that need to be set. So I expect kind of 80% to be done in the ground configuration setup, but then we'll add little pieces to configure along the way, which an example of that could be to add your more custom plate map, because the rest of your lab automation workflow is maybe reliant on a certain plate layout that we cannot suggest. So maybe that's where some of those more customized things come in.
All right, thank you. And May talked about the beta access. I will be sending the recording of this event to everyone who attended or who couldn't make it. And in there, we're also going to put a link to a form where you can sign up for early access, and then we'll reach out to you. So if you want beta access, just enter your email in that form. Moving on, and Magnus, pick up your application hat. Where do you see, from an application side of things, where do you see this fitting into existing workflows?
Yeah, I think a lot of sort of like stuff to define up front, but I think also showing the sort of vision of the whole workflow is super important, right? I think it's one of the big struggles that is very present always is, for example, analyzing the data or having that context that we talked about is super difficult if you don't do a little bit of work up front to write down things in a structured way. And so I'm really excited to just start working with that and seeing how that plays out. I think it is always something that seems a bit like a thing you're supposed to do when you're setting up an experiment, but it's one of the things that is difficult to really do well enough. And even if you do it, you still end up with the nightmare Excel sheet in the end, right? So I think that pattern is really common across lots of assays, and so I'm also just excited for us to start testing it out in the Reshape lab as well. Yeah.
Cool, thank you. And May, from more of a product and data perspective, where do you see it fitting into people's existing workflows?
Yeah, I think that depends a lot on how satisfied you are with existing workflows. There's different ways to integrate this in your workflow. You could go for a kind of replace the whole thing approach, whereas kind of seen in the video, you can just start from scratch and generate these experiment parameters, plate maps, and get your analysis done that way. If there's a scenario where you, for instance, store some information about your treatments, your compounds, your samples already in a LIMS, and you just want to bring in those entities into Reshape and then design your experiment, that's also possible. Or finally, if you're actually quite happy with the way your experiment design works, for instance, in LIMS, you can also just do the experiment design there and send the entire plate map with all its contents to Reshape. And then you run and you still benefit from seeing all of that content when you analyze the plates.
All right, thank you. And guys, I see your answers. I just have one more question for Magnus, and then we get to the questions in the chat. Magnus, this will work differently for different types of organizations. Could you talk through some of the organizations that we already work with and how it would fit into their workflows?
Yeah, for sure. I think this also plays into
how to work with this feature.
Yeah, for sure. I think that also plays into what Emma just said about the flexibility, first of all, right? I think some labs are extremely advanced in how they do this already, and maybe just looking for a really streamlined interface with Reshape. I think that's great. I think for a lot of cases we see, it can also be something where, maybe the workflow today is quite painful, right? So I'm really excited to see people start exploring this. I think in terms of types of organizations and companies, I'm really excited about the whole spectrum, honestly. I think for universities, for example, lots of academic users we have, at least, people are joining projects, leaving projects. I think simple things there, like really having the context nailed down and having these shared repository of understanding what's going on in itself will be super helpful, right? And also I think something that we touch on a lot is like helping make things more reproducible. For example, helping actually capture all this information that might be lost. So I think that's super exciting on one end of the spectrum, the sort of academic end. I think on the enterprise level, again, flexibility, super key. I think some companies might want to even do things like have anonymized treatments, but they can still use a lot of the functionality to actually streamline the workflow and send things back and forth. Use it to collaborate internally. I think that whole like automation, as a lot of companies are trying to upscale how many experiments they can do and streamline that whole process, I think will be super valuable for them. It's a desire we see a lot, people trying to get more done. But I think even these things like the monster Excel sheets is one of the big things that we see holding people back, at least. And so I think at that end of the spectrum, I'm really excited to see how people will really, be doubling down on more complex things like designing experiments and really scaling things up with that streamlining you get from something like this. So
cool. Thank you. And now on to the people online. Toge had a couple of questions actually, but I think the first one was: is there a human out of the loop feedback and the analysis to insight in the experiment planner? And can it provide suggestion on experiment 2 based on experiment 1 without going through human knowledge feedback?
Yeah, I can take this. I love this question because it's so spot on with what our vision is. So I think the story is coming across exactly well. It's not there yet. It's a vision, but it's definitely what we are aiming for.
Cool. And yeah, either one of you pick this up. Toge had another one, or you can also see it over there. But Toge asks about how to go from experiment design in like the software definition, what should be in each well, and then the physical plates actually be in the imaging device and ready to run.
Yeah, I can go for this one. So again, flexibility I think is the key. I think again some people will have pipetting robots and sample prep automation. They can utilize this also again through the API, for example, to send instructions back and forth and collate things. I think honestly a lot of people will use it manually, and there'll be some of these guiding tools such as, for example, the simple reagent calculations, like seeing the plate maps visually and preparing the samples by hand. So I think in that sense it can be both. Generally, I would say trying to build a very modern platform with a lot of integration possibilities. So I think for people who have those visions, it is also going to be something that would support those kinds of things.
All right. And then we had Jerome with a question: how does the AI know what to suggest?
Yeah, and I guess maybe we have a few questions.
Is there a bundling of questions going on?
I think we can maybe I can take the Jerome's question and also Daniel's after. So in terms of suggestions, this is going to be also exploratory, I think it's fair to say. We have a lot of interesting projects internally in the lab, as well as partners we're working with to generate some of these data sets that we're using to train a lot of these models. And so I think most people who use the platform today also see a similar thing with the image analysis model, right? So it's a key part of what we're also investing a lot in Reshape is building this data architecture to train these models that can predict interesting things. And if I can tie that directly with Daniel's question as well. So the question is, what about confidentiality of the data as well as the metadata? Who would have access? And will the Reshape team have access to experiment results and insights? So yeah, I think first of all, it's similar to the platform today in the imaging side of it, right? So of course, everything is confidential and everything is owned by the users. I see a few different options. Again, for enterprises, I think a lot of people have already expressed interest in anonymizing things, right? So it's completely compatible with, having anonymized treatments, for example, where, again, you put in a treatment, we don't need to know what it is, and you can analyze it, you can get all the insights and pull it back out. So that's one thing I think a lot of people will use. I also think a lot of people will use it where they do share the metadata. And so select people from the Reshape data science team, for example, are working with customers continuously on, designing these assays and implementing things. So yeah, very similar to the platform model we have today, I would say.
All right. And then Toge had another question, and I'm going to be the marking guy in the room. I am the marking, but DOE. Yes, what do you think about DOE? And I actually don't know what DOE stands for.
It stands for Design of Experiments.
There we go.
I think at least.
We never
and
know.
I think
So we correct if we're wrong.
yeah. And I think it's a very, I think it's also what the question alludes to is like this, really sort of large scale, systematic, statistical exploration of condition spaces and how you can do that. And I think I would say in the main use cases for the Reshape platform, I think people are typically not doing these very, very large, very systematic studies. I think, and that's what DOE really, I think, typically is used for, and really optimizing how you approach a problem. I would say this is really, I don't think I would say this is that, but it's like it's a step in that direction on helping bridge the gap from, again, these very manual workflows at very low throughput and difficult to scale up into something that's more structured, right? And I think, again, the power users of a system like that might combine it with a statistical DOE tool to, really be efficient about doing large scale experiments. But I would say it's, more something that some people are experimenting with, right? I don't think it's the typical thing people are doing today. So we're really trying to make it more user centric, making it more easy to use, making it something that's really for people who, are not happy with the workflows they have today around this. I think more than the really grand scheme of experimental design.
Maybe just to add to that, I think also design of experiments as a concept it has its limitations in two ways. I think thinking you have all of the relevant parameters up front is a limitation, and I think that's where machine learning and AI can provide a lot of interesting additions, suggesting new things to experiment with that you maybe didn't think about with your own context. I think the other limitation is the conception that you need to run all the experiments to get your conclusions. Maybe you have run very similar experiments in the past. Maybe there is strong knowledge already about certain behavior of combination of ingredients, so to say. So I think if we take those two things not as a given, but we keep flexible and we use ML and AI to fill in some of those gaps, I think that's where we can get to a really interesting place.
I was just talking a little bit here on this topic, but I think also one other thing, I think there's a lot of applications generally for this kind of tool, right? Some people might just be looking to characterize strains or just whatever it might be. Some people might be looking for the needle in the haystack or looking to sort the needles in the haystack. And I think, those different things also are very different. If you're characterizing a big set of strains, for example, I think, again, having a very structured way to set that all up front is going to be super helpful for people. So I think exploring the use cases is also a key part of it for us and seeing where it'll go with those first users, especially. Yeah.
Right. And Jerome just came in with the last question here, or keep them coming if you have more. We still have 7 more minutes. But he asks if you can automatically build the strain libraries with the experiment designer.
So take it.
Yeah, you can if you want to little bit of interpretation, I think, as well.
Yeah. I think essentially a version of your strain library is what you automatically be building. As soon as you put information about your experiment in, even already today, you can add it in the well details. So you can start building that up. But with the experiment designer, of course, you'll build it up more at scale. And we definitely also have it envisioned that we give you back this information. So you can easily go to all of your experiments that you've previously run with a certain strain or a certain organism and get your insights on that level and not just on an experiment level.
I think it's actually something I'm personally quite passionate about, and we work actually with some strain collections because I think if you work with microbial work today, if you look up a strain, it'll typically say grows in media X, and that's really often all there is, right? I think a version of this is where you actually can start in a structured but also easy way collect actual data on, okay, these strains actually grow well in this whole range of things, so they grow exactly this quickly in these conditions or whatever it is, right? And being able to explore all that. So yeah, I think it's one of the, again, just the characterization side, I think is a really key kind of use case for this.
All right. And Tugur came in with another question on what types of use cases we're mostly excited about for the experiment designer and what would be developed or released first.
Yeah, I think essentially we're most excited about the use cases that you are excited about as well. So I would love to get in touch and learn about that. I think, as I mentioned in the beginning of the session as well, I'm personally very excited to see if we can bring some more efficiency, like a bigger scale, more complexity into workflows with this designer, because it all becomes a bit more manageable to handle all that data. So I'll be definitely looking in that direction. But yeah, there's many opportunities.
Yeah, I really mirror that. I think it is like, this is one of the key, I think, things holding people back from going from small experiments, if you could call it that, where it's like really limited in conditions, to something that's like a mid-scale thing, right? And so I think lots of experiments fit that bill, which is also part of what's exciting, right? And I think, yeah, I'm most excited about seeing people try to sort of ramp up and like get out of the typical practical limitation of, really having to nail down exactly which conditions you want to test, but having more this maybe exploratory view of it, where you actually have the resources to do more things.
All right. And that was actually, we don't have any more questions. We'll, of course, Togu came in with, just before we close. That's great, Togu. We still have 4 more minutes. But in the setup, there was a lot of reagents, sorry, specific antibiotics. Does the system know what that is to provide this metadata? And can the user create a custom compound and enter metadata about it and its properties?
I think a combination of the two. We're doing a lot of experiments in our own lab. We're building out a lot of data. Of course, there's also public knowledge about some of these compounds, but it will also be able to kind of build up your own collection. And that's where, of course, we don't have the data up front, but you can kind of build up while you're experimenting and running more jobs in the Reshape platform, kind of build up that knowledge specifically for your organization only.
And I think also we do provide a lot of like options. For example, there is some searchability, so you can quickly find the right things, but we also do allow completely custom things, right? So if you just want to say we have mix A and it's a whole complex media recipe with a lot of stuff, that's fine as well, right? You just have to know that mix A is the level of detail you can go into when you're analyzing your things at the end, right? So we talked about flexibility a few times. I think it's really one of the key things in R&D environments, right? It needs to fit a lot of use cases. So.
All right. And now I'm actually going to shut off for questions. We'll, of course, be willing to respond to anything if you have more that comes up when you rewatch this event. Just to reiterate, we're going to send you the recording of this event with the beta access form, so you can use that if you want to play around with this when it's live. But yeah, just let us know if you have anything more that pops up when you rewatch this. And that was it. Thank you guys for tuning in. We hope you're excited about it. I know you guys are.
Yeah, very.
Yes.
Thank you for the questions as well. It's always good. And yeah, super excited to follow up on this. That'll be super fun.
Yes.
Cool.
See you guys.
Have a good day.
Bye.